W.B.Langdon, an RA in the CREST group. What I wanted to talk about is using genetic improvement to improve a particular program, RNAfold. So I'm going to start by introducing RNAfold. What we've actually done, the title talks about two things. Firstly is speeding up RNAfold, but mostly I'm going to talk about functional improvements. So most of the work up to now on genetic improvement has been improving non-functional aspects, principally things like speeding up or reducing resource consumption. Here I'm going to talk, and there the goal is to take the same program and produce answers faster. Here I'm going to be talking about taking a program and producing better answers. The work on speeding up was presented at the GI workshop at Gecko in July, and since then we've done a small amount of work on improving RNAfold by changing its code. Most of what I'm going to be talking about is improving RNAfold by changing constants with units. And the next thing we might look at is going back to speed up, but instead of using 128-bit SSE instructions, using 512 SSE instructions. So hopefully that will be a straight follow-on from the previous work. But as I said, most of the time I'm going to be talking about optimizing the parameters within RNAfold. So RNAfold is a piece of C code. It's about 7,000 lines of code. It's part of the Vienna package. So this is state-of-the-art code. It's in use by many bioinformatics teams, including the ETRA group, who have used it for modeling RNA on a global basis. So you can get hold of an app which internally runs RNAfold. And we've had contact with that group, and they're now using the SSE version, at least internally. What RNAfold does is it takes a sequence of RNA molecules, RNA bases, so represents a molecule as simply its base sequence, and then predicts how it will fold up. So this is actually an average guy from RNA strand, and you can see its prediction for this part of the molecule is pretty good, but overall it gets a correlation coefficient of just over 0.5. So there is quite a lot of scope for improvement. And what we're going to do is we're going to run RNAfold on data from this other database, RNAstrand, which not only has the RNA sequences, but it actually has, as far as it's known, the correct secondary structure. So this structure is believed to be actually the correct structure for this particular molecule, SRP00287, which is a molecule that is believed to exist in every living cell on the planet. So the way RNAfold works internally is it uses dynamic programming to predict the energy of different ways of folding up the RNA, and simply takes the folding structure which has the lowest predicted energy. It scales approximately as n cubed, where n is the length of the molecule. Internally, it has over 50,000 parameters, which essentially represent estimates of the energy of interaction between different parts of RNA, different bases within the RNA molecule. And what we're going to do is use GGGP, in other words, the grow and graft variant of GP, to optimize those 50,000 parameters. And when we, for each time, come up with a new set of parameters, we run RNAfold on a collection of molecules, and we compare the predicted structure with the actual structure in RNA strand. We're going to use 1/3 of the molecules for training, and for reasons of speed mostly, we're going to just use training on the shortest molecules. And in the experiments I'll describe, we actually used all 681 short molecules from the training data. In practice, it might have been better just to subsample that 681, but the program can run through those in about a second, so it was left at using 100% of the available training data. We're not going to, although we're going to use essentially a GA, what we're going to do is, instead of representing all 50,000 parameters as a bit string, which would be about 1.6 meg, what we actually do is we represent the genotype, so there's a mapping from genotype to phenotype, and then each of the genes within an individual is applied to the data structures one at a time, and then we move on to the next one. There are multiple types of mutation, which I'll describe shortly, and we also use 2-point crossover. So the three main types of mutation are replace, which what it actually does is every instance of a particular value within an array is replaced with another one. So here in the top example, every time there's an element in this array whose value is 260, it's replaced with 80. So this potentially makes a large number of changes. And as you can see, this is much more compact representation than changing explicitly all the array indices in 22. The second way of doing it is perhaps more intuitive. You simply replace a given element of an array with a new value. And here we're using wildcards. Thank you. So in this example, we replace all of mismatch I for the first two indices, and where the third index is 0 by 100. And the last example is we simply, instead of replacing with an absolute value, we change the current value. So here we're changing the current value by adding 20 to it. Most of the parameters within RNAfold are representing energy values, and those are always multiples of 10, or this strange value INF, which basically is used internally to mean infinity. But there are a small number of arrays that have small values, typically between 0 and 8. So although the examples I've got here are multiples of 10, you can also have smaller values. And the last mutation operator is, after we've started using these, we can actually apply a mutation to the mutation itself. So we could change that 80 to a 70, for instance, or to a 90. And we do that with this Greek mutation. So the way the fitness works is we're going to run our version of RNAfold with different parameters. It produces a new, different predicted structure for each of the molecules, and then we're going to compare that with the actual structure in RNA strand. We also compare against the previous version. So if the mutation didn't make any difference, then we ignore it. Otherwise, we calculate fitness, which is this correlation coefficient, and we sort the population by average fitness value. So we take the top half of the population and give each of those 2 children. And yeah, if there are things that, if we tweak the data structures in a way that causes the program to segfault, then it can't be apparent. So evolution, half mutation, half a crossover, various operations to try and promote search. So for instance, once we've tested a particular genotype, we prevent that genotype being reused later. And we've also got this anti-elitism fitness function, so that the population can't cluster around a given fitness value and stay there forever. We've got a somewhat arbitrary limit of 1% of the population size, so that if a particular fitness value is being used more than 20 times already, then that particular individual is not going to be a parent, not going to have children in the next generation. We run for 100 generations and a population of 2,000. And so this is just a plot of fitness. So fitness is this Matthews correlation coefficient, which is guaranteed to be between -1 and 1. The unmodified system starts down here at 0.66, and you can see over time the best in the population actually improves over time. What this graph does not show you is the size of the individuals in the population, which bloats enormously. So by the time we get to this last guy, it's about 2,000 individual mutations long. And so we do our usual trick of going through the individual and dropping out bits of it one at a time. And if removing that part makes no difference, then we remove it permanently. And that very simple hill climbing operation actually reduced the length from over 2,000 separate mutations down eventually to 42. And makes a very marginal difference, actually a slight increase in the correlation coefficient on the training data. We take that individual and we try it on the test data, the holdout data, that the GP is never seen. So on the data it's never seen, the performance is pretty much the same. This is the performance on the short individuals, so individuals like those that it was trained on. So essentially it's not overfitting. This is a summary of the 42 changes. I'm not going to go through all of them, but maybe I could just start with the top one, which is the easiest to explain. In red, we've got the name of the array that's been mutated. That operation simply means replace everything that was in that array with 560. Surprisingly, that is sufficient to actually improve the performance of the whole program. And you can see there are some other ones which are much more complicated, and there are also other simple ones like this one, which just replaces the current value with the value 80. And this surprising one, which takes all the current values, which are 70, and replaces them with infinity. Here I've just sorted the arrays by how much impact they have. So some of the changes I've just shown you actually don't make very little difference at all, and some of them make quite a large difference. So this is a somewhat disappointing graph. I should be honest about that. What we've got plotted horizontally is the original performance of RNAfold. Again, on this Matthews correlation coefficient. And if you believed the original hype about RNAfold, then everybody would simply score 1. Everybody would be up here. But in reality, the original code is scattered all the way from 0 up to 1. There's even actually a few slightly negative ones. If I'm going vertically, we've got the performance of the mutated code. So essentially, everybody in the upper triangle is doing better than the original one. unfortunately, there are also guys in the lower triangle which are actually doing worse. But overall, there are 769 cases where the mutated code is doing better, and 460 where it's doing worse. So it's a bit of a split decision, but it's definitely, on average, doing better than it was before. And the difference between those two is 19.89%. So what I'm going to show you next is two examples where it does reasonably well. This is the original code. So this is the RNA sequence. This is the RNA sequence, how it actually folds up in reality. So that's the original prediction, and this is the prediction of the new mutated version. So you can see, essentially it gets it all right, except it doesn't predict a binding between the C and Gs there, whereas this one actually has got a binding there. So that's a fairly short molecule. This again is another example where it does well. This is the original prediction, and as you see, it is actually correlation is actually slightly negative. So that's the actual structure from RNA strand, and the correlation is 0.85. And you can see that they pretty much lie on top of each other. There is this non-standard binding between the C and the A there, which RNAfold knows nothing about. That's one of the special types of binding which is essentially left out of most of the elementary textbooks on DNA and RNA folding. Okay, so to summarize, what we've done is we've applied GGGP to a state-of-the-art RNA folding tool. In one case, which is the example we talked about earlier, we got an overall 31.9% speed up by changing the code to use SSE instructions. Unfortunately, only the manual part of that change was accepted by the owners of RNAfold, but nonetheless, they did take part of the changes. And when I checked before we went to Gecko, that version had been downloaded over 2,000 times, and it is in use by the eTRNA project within their internal development. Since then, we've looked at not just speeding up RNAfold, but also improving its predictions, and that's what I spent most of my time talking about. So overall, we've got a 20% improvement by mutating the parameters within the code, but not actually changing any of the code at all. And at that point, I should really stop. Thank you.